I'm Kenny.
And I'm Noor,
We are from International School of Kuala Lumpur.
and we're gonna introduce the cell theory.
The cell theory also states that the cells are made from pre-existing cells. Cells are the smallest units of life. All the living things are composed of cells, and we can find cells anywhere in our life. Plants, human, and dogs. The cell theory said all cells come from pre-existing cell. To prove that, we give an example just like the babies are all made of pregnant women which are already pre-existing cell. Since cell is the smallest units of organism, we can't see it through our eyes. We need something called microscope. Microscope allowed us to observe that all living things are either unicellular or multicellular. One of the functions of unicellular organisms carry out was reproduction. It produced offspring either sexually or asexually. As you can see, the picture illustrates that the molecule has the smallest size. It had got 1 nanometer. And membrane, it's about 10 nanometer. Viruses are about 100 nanometer. Bacteria are 1 micron. Organelles are up to 10 microns, and most of the cells are about 100 micron. In order to calculate the linear magnification of drawings to the actual size of the specimen, the magnification equals to the size of the image divided by the actual size of the specimen. Drawings of microscope structure must include at least one of the scale bars or magnification numbers. Surface area to volume ratio is very important to cell. As the dimension of a cell increases, volume increases proportionally faster than surface area. Thus, the surface area to volume ratio decreases with cell size. Setting up an upper limit on cell size because lower relative surface area reduces rates of change, while higher relative volume increases metabolic demands. Thus, the rate of exchange can meet the needs of metabolic demands. By calculating the volume ratio to surface area, you divide the surface area by volume. Then you get the answer. Multicellular organisms are said to show emergent properties because of the whole organism is more than the sum of its parts, caused by the complex interaction between cells. Cell differentiation in multicellular organisms ensures that every function required by the body is able to occur. For example, nerve cells are specialized to transmit information. Red blood cells are specialized to carry out oxygen. If no cell differentiation occur in multicellular organisms, all cells would be the same and no specialized cell function would occur. Stem cells are cells that are found in all multicellular organisms. They are characterized by the ability to renew themselves, which is have the capacity to divide and self-regenerate through mitotic cell division and differentiate into a diverse range of specialized cell types. There are many therapeutic uses of stem cells. One of them is the bone marrow transplants. They only work because when you're actually transplanting the hematopoietic stem cells into the bone marrow, and the peripheral blood stem cells as well as the cord blood stem cells can be used in lieu of bone marrow, making being of donor FAR easier today than in the decades past. As you can see, the picture is a prokaryotic cell, and the cells are used to protect cell from outside environment and prevent cell from bursting internally. Plasma membranes are semi-permeable membranes that controls the substance from moving in and out of the cell, and it contains peripheral proteins and substances pass through it by either active or passive transport. Cytoplasm contains many enzymes and used to catalyze chemical reaction of metabolism and DNA. Pili are used to help bacteria to adhere to each other for the exchange of genetic material. Flagella are made of proteins called flagellin, and it helps bacteria to move around by the use of a motor protein that spins the flagellum like a propeller. ribosome, they are where the protein synthesis occurs, and it contributes to protein synthesis by the translating messenger RNA. Nucleus, it is the region who contains the DNA genetic information and controls the cell, and it will be passed to the daughter cell. Prokaryotic cells divide by binary fission. Binary fission is a method of asexual reproduction involving the splitting of the parent organism into two separate organisms. As you can see in the picture is a eukaryotic cell, and the ribosome are found either in the cytoplasm or attached to the surface of rough endoplasmic reticulum, or in mitochondria or chloroplast. Ribosomes are the site where protein synthesis occurs as they translate messenger RNA to produce protein. The rough endoplasmic reticulum can modify proteins to alter their function or destination. Synthesized protein to be excreted from the cell. Lysosome contains many digestive enzymes to hydrolyze macromolecules such as protein and lipids in their monomers. Golgi apparatus receives protein from the rough endoplasmic reticulum and may further modify them. It also packages the protein before the protein is sent to its final destination, which may be intercellular or extracellular. Mitochondria is responsible for aerobic respiration, and it converts chemical energy into ATP using oxygen. Nucleus contains the chromosome and therefore the heterogeneous material. The genetic material is responsible for controlling the cell. Comparing prokaryotic cell to eukaryotic cell, prokaryotic cell have naked DNA which is found in the cytoplasm in the region which is called the nucleus. On the other hand, eukaryotes have chromosomes that are made up of DNA and proteins. These chromosomes are found in the nucleus enclosed in the nuclear envelope. Prokaryotes do not have any mitochondria where eukaryotes do. Prokaryotes make use of their cell surface membrane and mesosomes instead. Prokaryotes have small ribosomes compared to eukaryotes. In prokaryotes, there are either no or very few organelles bound by a single membrane, compared to eukaryotes, which have many of them, including the Golgi apparatus and the endoplasmic reticulum. The difference between plant and animal cells is that animal cells only have a plasma membrane and no cell wall, where plant cells have a plasma membrane and a cell wall. Animal cells do not have chloroplasts, where plant cells have them for photosynthesis. Animal cells store glycogen as their carbohydrate resource, where plants store starch. Animal cells can change shape due to the lack of cell wall and are usually round, whereas plant cells have a fixed shape kept by the presence of the cell wall. The two rows of extracellular components are the plant cell walls. It gives the cell a lot of strength and prevents it from bursting under high pressure, as it is made up of cellulose arranged in groups called microfibers. It gives the cell its shape, prevents excessive water uptake by osmosis, and is the reason why the whole plant can hold itself up against gravity. The animal cell contains glycoproteins in the extracellular matrix, which are involved in support movement and enhancement of the cell.
This diagram shows the structure of a membrane. It includes phospholipids bilayer, cholesterol, glycoproteins, integral proteins, and peripheral proteins. In a protein membrane, the phospholipids have two parts, a hydrophilic head and a hydrophobic tail. The hydrophilic head means that the molecules are attracted to water. The hydrophobic means that the molecules are not attracted to water, but attracted to each other. The phospholipids are stabilized by the hydrophilic properties. Fluidity helps the membranes to be stable. The structure helps endocytosis and exocytosis to occur. The The hydrophilic and hydrophobic properties keep the phospholipids from moving. The membrane proteins have many functions. These include hormone binding sites, immobilized enzymes, cell adhesion, cell-to-cell, communication, channels for passive transport, and pumps for passive transport. Passive transport requires both diffusion and osmosis. Diffusion is the passive movement of particles from a region of high concentration to a region of low concentration. Osmosis is the passive movement of water molecules across a permeable membrane from a region of lower solute concentration to a region of higher solute concentration. Both facilitated and simple diffusion, no energy is used to cross the membrane. However, in simple diffusion, the molecule is usually small and hydrophilic, so it can easily get through the phospholipid bilayer. In facilitated diffusion, the molecules are usually large in size and they are hydrophobic. Because of their size, they go through the transport proteins. Now I'm going to explain the role of protein pumps and ATP in active transport across membranes. Active transport is when substances are transported across a membrane by using a form of energy called ATP. Active transport can let substances move against the concentration gradient. Protein pumps are found in the membrane. They are used for active transport. Each protein pump is responsible for transporting a substance. This is because cells need to be able to control what is absorbed and what is moved out. I'm going to explain how vessels are used to transport materials within a cell between the rough endoplasmic reticulum, Golgi apparatus, and plasma membranes. Vessels are made by taking off one piece of membrane. This happens because the membrane is fluid. Vessels are used to transport contents to the cell or inside of the cell. Proteins are transported to vessels from the rough endoplasmic reticulum to the Golgi apparatus. The making of vessels from the plasma membrane let materials be taken in. Phagocytosis, pinocytosis, and endocytosis is when the contents are taken in using a vessel. Exocytosis is when contents are secreted using a vessel. I'm going to describe how the fluidity of the membrane allows it to change shape, break, and reform during endocytosis and exocytosis. In endocytosis, a fraction of the membrane is pulled in. Some of the fluid becomes with this when a vessel is taken off. Part of the plasma membrane is pulled inwards. Vessels can then move through the cytoplasm by carrying material. In exocytosis, vessels are combined with the plasma membrane. The material inside the vessel are then taken out and the membrane levels out again.